For the sake of this video, we will define biotech as the exploitation of biological processes for industrial or other purposes, especially the genetic manipulation of microorganisms for the production of antibiotics, hormones, etc. Humanity, for the longest time, lived with little control over the natural world. That is, until around 11,000 BCE, when the first known plants were domesticated in the Middle East in an area now known as the Fertile Crescent. This idea spread around the world. The process was done with animals as well, resulting in the start of selective breeding, which is defined as the intentional breeding of organisms with desired traits in an attempt to produce offspring with similar desirable characteristics or with improved traits. This led to pigs that oink, cows that mooed, and broccoli that stood by silently. This also led to the creation of dog breeds. In the new formed civilizations of Egypt and Mesopotamia, great progress was being made in the field of biotech. That was the discovery of fermentation. This led to the creation of beer, wine, and cheese. Though the process was not fully understood until the 1850s, with discoveries made by Louis Pasteur. In 1663, the first recorded description of living cells was made. By 1675, bacteria and protozoa were discovered. In 1798, the first viral vaccine was used for smallpox. In 1838, protein was discovered, named, and recorded. Mendel discovered the laws of inheritance in 1863. The following year, the first centrifuge was used. In 1869, DNA was identified in the sperm of trout. In 1885, the first rabies vaccine was used. In 1917, a pure microbiological culture was used to produce acetone, which was needed to produce explosives in World War I. In 1919, we see the first uses of the word biotechnology. In 1928, Alexander Fleming discovered the mold penicillium. In 1933, hybrid corn was commercialized. In 1950, the first synthetic antibiotic was created. In 1953, Watson and Crick described the structure of DNA. In 1972, genetic material was transferred into bacteria. In 1990, the first federally approved gene therapy treatment was performed successfully. In 1996, the first mammal was cloned from an adult somatic cell. In 1994, the United States Food and Drug Administration approves the first genetically modified food. In 1999, the gene responsible for cystic fibrosis was discovered. And in 2003, the Human Genome Project was completed.